Hello and welcome. In this video, we shall see the scanning electron microscope. We shall see all the details, that is principle, then the instrumentation and the working and different images, as well as what are different applications of scanning electron microscope. So first of all, this first scanning electron microscope was developed by Ernest Ruska in 1933, and he received Nobel Prize for this discovery in physics in 1986 along with G. Binning and H. Rohrer. G. Binning and H. Rohrer, they got this Nobel Prize for scanning tunneling microscope, that is STM, and it was shared with scanning electron microscope discovery for Ernest Ruska also. So all of them, they received the Nobel Prize in sharing in 1986 for physics. Now we shall know what are the limitations of an optical microscope before going into detail of an scanning electron microscope. Now, according to Rayleigh criterion for microscope resolution, we have this capital R is equal to lambda divided by 2n sin theta, where this numerical aperture is taken as the product of n multiplied by sin theta. This half of the lens angle is called as theta. The refractive index of the medium between the specimen and the lens is the small n, and the wavelength of the light emitted is nothing but what? Lambda. Now, in most of the optical microscope, this n into sin theta product, so we call it as what? Numerical aperture. So we get it capital R is equal to lambda divided by 2n. Now this product n into sin theta, that is numerical aperture, is generally equal to 1 because we cannot increase this refractive index indefinitely. It has certain limitations, and also this sin theta is also having certain limitations. So therefore the product is generally equal to 1, n into sin theta. So we get the formula for resolution as lambda divided by 2. Now, in case of an optical microscope, we know that we use nothing but what? Visible light. The range of this visible light is 400 nanometer to 700 nanometer. So we will consider one of the wavelengths, say 500 nanometer, in this visible range. So therefore, the resolution of an optical microscope will be lambda divided by 2, which is 500 divided by 2 nanometers. So it comes out to be 250 nanometer. If we convert this into micrometer, we get it as what? 0.25 micrometer. Now, with the help of an optical microscope, we can go to the order of few micrometers, of the order of micrometer only. We cannot go beyond this, that means what? The below range, in nanometer range, we cannot go, we cannot see or observe with the help of an optical microscope. So therefore, we need some microscope which has better resolution, say of the order of nanometers, to observe the nanomaterials and some other objects which are of the order of nanometer size. So for this purpose, the main limitation is the wavelength, which is visible light. So if we use, instead of visible light, if we use the electrons, that is the electron beam, then it becomes possible. We'll see how this becomes possible. Now, according to de Broglie hypothesis, we have lambda is equal to h divided by p, where this h is the Planck's constant and p is the momentum. And the kinetic energy, which is a non-relativistic kinetic energy, is given by small e multiplied by capital V. This capital V is the voltage applied to the electron gun, is equal to half mv square, that is the kinetic energy. This m is the mass of electron and v is the velocity of the electron. Now we will multiply this equation 2 on both sides by the small m. Then we get this as MEV is equal to half m square v square, or by rearranging the terms we get 2 MEV is equal to m square v square. The product of mass and velocity is momentum, so m square v square is nothing but what? P square. So if we find out momentum here, we get momentum is equal to P is equal to square root of 2 MeV. Now we will substitute this in equation number 1. This equation 3 we will substitute in equation 1. So we get here that lambda is equal to h upon square root of 2 MeV. Now this h is the Planck's constant, which is 6.626 into 10 raised to power minus 34 joule second. M is the mass of electron here, which is 9.109 into 10 raised to minus 31 kg. E is the electronic charge, which is 1.602 into 10 raised to power minus 19 coulomb. By substituting all these values over here, we get this wavelength is equal to 1.22 divided by square root of the applied voltage to the electron gun and this much nanometer. So we get this wavelength in the nanometer. Now we will derive one more formula to find out the relation between energy and the wavelength in nanometer. So we know that E is equal to h nu and we know that C is equal to nu lambda. So from here we get nu is equal to C divided by lambda. So we can write down this E is equal to h nu as E is equal to hc upon lambda. Now substituting this Planck's constant and the velocity of light here and then dividing that by 1.602 into 10 raised to minus 19 to get the energy in electron volt. So we get this relation between energy in electron volt is equal to 1240 divided by wavelength in nanometer. So we got these two relations, that is relation between wavelength and the applied voltage here and relation between energy in electron volt for a particular wavelength. Okay. So now by substituting these different voltages here, say if we substitute here voltage, this capital V is equal to 20 kV, that is 20,000 V, 20,000 volt, then we will get this lambda is equal to 0.0086 and corresponding energy in electron volt we can find out with the help of 1240 divided by this lambda in Nm, that means 1240 divided by 0.0086, we will get this energy in million electron volt. Similarly, or million electron volt or mega electron volt we can say. Similarly, for 50 kV we will get this wavelength and this energy, and 80 and 100 also. Now here we have this different graphs actually just to explain. So this graph shows the wavelength versus the applied voltage. So as the applied voltage increases, we see that applied voltage increases, the wavelength goes on decreasing. And there is other graph, that is energy versus wavelength. So as the wavelength goes on increasing, the energy goes on decreasing, and as the wavelength decreases, the energy increases. So they are inverse, because we can see in these two relations they are inversely proportional. Of course here lambda is inversely proportional to root of V, and here energy is inversely proportional to wavelength. Okay, so this can be seen in these two graphs here. Now if these are the wavelengths, then what will be the resolution? It will be capital R is equal to lambda by 2, so which is nothing but point, say for this first one I will say for 20 kV, we get it as 0.0086 divided by 2 nanometer, which comes out to be 0.0043 nanometer. Now remember, this is the theoretical value. Actually there are so many other parts in scanning electron microscope which plays the role in determining the resolution of an scanning electron microscope. So the resolution of an scanning electron microscope is generally between 3 to 10 nanometer only. But this is the theoretical calculation, that means we can go maximum with the help of this theoretical calculation we can go up to this. Okay, but in actual practical it is not possible to get this resolution, because there are so many other factors we will see in the instrumentation of a scanning electron microscope. So that means what, if we use this wavelength, then we can have a better resolution rather. Okay. So this is the construction of an scanning electron microscope. So this first one we have the electron gun. Now this electron gun continuously emits these electrons. Then Now in case of an optical microscope, we have different lenses here, but here we cannot use the lenses. In case of a scanning electron microscope, we have to replace those lenses with the electromagnets, big electromagnets, so they are called as the condenser lenses. And to scan these on the surface of the sample, we need scanning coils, and these things are controlled with the help of a computer nowadays, with the help of a computer, and which will generate, this scan generator will generate a raster pattern. Now what is raster pattern? It is nothing but the similar pattern which is there in the old television, cathode ray television, cathode type of television, which have the picture tube, etc. So that type of scanning done, that is called as the raster pattern on the surface of this sample, and there will be different emissions once we make these electrons incident on this sample. Then out of them, the selected emissions are only detected here, and after amplification they are given to the computer to generate the scanning electron microscope image. Now when we make this electron beam incident here, there are different types of emissions here. These are shown over here. Out of all these emissions, only the secondary electrons, backscattered electrons, and the characteristic X-rays, these three are important. Out of these three, only these two are important for generating the image, and this is characteristic X-rays are used for elemental analysis. We will see some graphs regarding that also in our coming discussion. And from the different parts of the sample, these emissions are coming, that are shown here in this. So here in case of SEM, we have to concentrate with these two for image generation, and this one for the elemental characterization, that is called as EDS or EDAX. We will see it in a later discussion. Okay. Now this is the scanning electron microscope. Now just now we have seen it here. So this scanning electron microscope system is kept in very high vacuum, say generally below 10 raised to power -3 Pascal vacuum is required, so or even better vacuums are required. So this total system is in high vacuum there, and here we have the sample chamber. Now in the sample chamber only we have this EDS detector is there for detecting the characteristic X-rays, and this is secondary detector and backscatter detector. These two detectors are used for generating the image here. Okay, so this is a microscope. I had taken it from this website. Now what is electron dispersive analysis of X-rays, which is called as EDAX or energy dispersive studies, that is EDS. X-rays emitted from the sample are collected by the detector, which requires liquid nitrogen for cooling, and the graph of intensity against energy is plotted. The peak is the characteristic of the material so that the material can be detected. The detector collects the X-rays from the sample. Now we will see what are the steps involved in preparing the specimen. So specimen preparation, mainly the following three aspects should be taken into consideration. The first one is remove all the water, solvents or other materials that can vaporize while in the vacuum. Then second step is there, that firmly mount all the samples. Then we have the third step, non-metallic samples such as bugs, plants, fingernails and ceramics should be coated so that they are electrically conductive. So metallic samples can be placed directly into the SEM, but for these non-metallic samples we have to make a coating. Generally we have to make a platinum or gold coating on the non-metallic or biological samples. Now here we have different SEM images. So this shows the SEM image of CEO2, that is ferric oxide, Syria we call it as, and this is the EDS of this. Now here you can observe that the peak of CE is approximately half the height of peak of oxygen. So this means that this is nothing but what? CE is present here as half of oxygen. Okay. Now in case of this EDS image, say like our elements having smaller atomic numbers like hydrogen, helium, they are very difficult to detect here in case of EDS because this is having some high energy. So those elements are difficult to detect here in case of EDS. Now this shows the CNT on the alumina substrate, that is the carbon nanotubes on the alumina substrate, and here we have the CNT on the silicon substrate. Then this shows the nano diamonds here. This is the magnified view here. Then this is the image of an ant. So actually it got the best prize here by the JSM. So he got this best prize for this high there image, so which I had taken it from this website there. Then this is the image of a bug with the help of SEM. These are the different types of pollens, image of different pollens. Then this is the image or the head of the leaf beetle is shown here in this image. This is the image of an algae. This is surface of the kidney stones. So like that we can have various images there. Now what are the applications? So there are various applications in scientific field, then in case of medical uses, then in forensics, and then in metal surfaces, to study the metal surfaces, etc. So these are the various applications. Now I will give some homework. Say find out the wavelength corresponding to 90 kilovolt, that is 90,000 volt applied to the electron gun. Then find the energy corresponding to 90 kilovolt, that is 90,000 volt applied to the electron gun. And find the theoretical resolution of SEM corresponding to 90 kilovolt applied to the electron gun. And one more thing, that compare the optical microscope with scanning electron microscope. I hope that you understand this. You can write down these answers in the comment below. If you have any difficulties also, you can ask me in the comment below. Do like, subscribe and share. Thank you very much.